Maniswini, invite your all friends to join.
Okay, sir.
Okay, so numbers are now good. So now we are about except one, all are here. Okay, so now we are at the module number 2, and in the module number 2 we have to talk two most important features. That is your hybridization and the probe. And as we talk about in our previous class also, that the principle of the diagnosis or principle of the finding or in case of DNA is all based upon the complementarity, because the unknown thing, we don't know what is the sequence in the unknown sample is going to be, but what we know is that the sample or the the thing that we are having. So like if you're having a sequence like AGTC that I mentioned in that case, if you're having that, then you can find whether that sequence is present in your sample or not. So this is that thing is going to happen on the basis of the complementarity. So that complementarity that we all know, that A pair with T and G pair with C, that we call the Watson and Crick base pairing. So this is the simple thing, and GC pairs more energy that is the simple that you all know about it. Nothing is new that I'm referring here. And this base pairing is happening as if your sequence is going to have more GC, then the binding will be strong, and less GC is going to be strong. So this is a simple 3 prime 5 prime. The green one is going to be 5 prime to 3 prime. That is going to run in the opposite direction, and 5 prime you are having phosphate, 3 prime you are having phos. This is a simple that we all every time know. Now it comes to this thing, how we are going to help, what things are going to help us is that is going to be probe and primer. So these two things are there by which we are going to utilize, which we are going to use for detection of the presence or absence of your DNA. Where we will detect, that is going to be talked about in our coming slides. But first thing, we should have our reagent ready. Then only we can move further. Otherwise, we can't move further. So first thing, what is a probe? So now, simple as I mentioned that the DNA sample that you are extracting in your practice, you mentioned that DNA you are getting from a cell, and we don't know what property of your or sequence your DNA that you are going to get have. So simply you can say that in our case, if you talk about, like if I'm having a gene uh, like a gene of hemoglobin, you can say hemoglobin gene, the globin gene that I'm having. So about the globin gene, we know that this is a sequence. Now the gene sequence is available with you. But if there is any change in that gene or not, that you want to check, is the same sequence that we are thinking is there or not. So that if you want to found, so that's for that you need a known sequence and an unknown sequence. So unknown sequence means that you are not aware about the sequence, whether that is present or not. So simply again, that is not a good at best example. I think COVID example is going to help you better in this way. So simple thing, if you say that like in our case that you are extracting, you are taking a nasal swab and a throat swab, and whether your sample is going to have a viral DNA, viral RNA or not, that is our aim to know. While RNA is present or absent, that you want to check. You extracted the RNA, but you don't know whether it is there or not. So how to find it? So if that you know the sequence, that sequence is there, you can design a probe for that sequence that should be present in your RNA if the infection is there. Okay, so that is a known sequence. So you make a complementary sequence of that. Means if your RNA is present in your DNA sample, if you are having a complementary sequence, as I mentioned in your previous class, that RNA can also be double-stranded, but RNA is not double-stranded. The reason behind it, if you don't have the complementary sequence within you. If you are having the complementary sequence, that may also become double-stranded. Okay, so if that RNA you want to check whether the RNA is there or not, so that you can find by taking another sequence. But both the one more thing is both the sequence should be single stranded. So here the two things, this is known sequence that we are designing. We are naming by two different names. One we are calling probe, another we are going to call the primer. So these probes and primer we are going to get name depending upon their utility, their size, that we'll talk and discuss and continue just a few minutes later. So as I mentioned you that the sequence that you are referring, that you want to know, is whether that sequence is present or absent. So like in case of, as I give you an example of your COVID virus, SARS-CoV-2. So SARS-CoV-2, we know that this is a sequence of SARS-CoV-2. So simple, if you say that, sorry. So yes, you can say that this is a sequence SARS-CoV-2. And we are with this SARS-CoV-2, we are going to have a large human genome, human DNA also. So like two samples are there. In one sample it is going to be there, in another sample it is not going to be. So this both are, this H is human, this is also human, this is human, this is human, and this is going to be also while in origin. So now our aim is to whether, because we can't see, just is an assumption that in these two samples, these two are present. One may be having it, another may not be having it. So, but you need to find it. So, how to find it? So, for that, this sequence, we know that if the virus is going to be there, the virus is going to have some DNA sequence like this. So, the A, G, T, C, T, A, G. So, this sequence is going to present the virus. So if you want to detect this sequence, what you can have, so like this sequence is going to present here, this A, G, T, C, T, A, and G. So this sequence is going to present in the virus. So now this is single stranded. It should be single stranded, and only it can pair with the complementary one. This DNA being double stranded, it is not going to pair. But if you want this sequence to present on the human DNA, also you want to detect whether the human DNA is going to have that sequence or not. So for that, we will talk a bit different way. So now this sequence is present in the virus. Now what we want to detect it? With the same virus sequence, we can't detect it because this sequence is, if you go there, it's not going to pair up. So no complementarity is there. But if you make a reverse sequence, like this A is there, so in A, you're going to have a T. For G, you need a C. Then you need A. And you need again a G. Then you need A. Then you need T. And then you need C. So this is a 5 prime, 3 prime. And this is going to be 3 prime and 5 prime. Okay. So, but when you're representing it by convention, as we all, when we're talking about our molecular biology, so if you just a convention is there, the sequence that we are going to write, it should always be in the 5 prime to 3 prime direction. Okay, so this sequence is going to be complementary with it. So what is going to happen? This pair will be T, C, A, G, A, T, C. Okay, so this complementary sequence is going to come here and it is going to pair up with it. So this is a simple thing that is going to happen. This is the principle that we're talking about, the principle of detection or diagnosis or the finding in our molecular biology or the molecular biology tools that we have. So both when you're referring, if you've seen primer and probe, both are talk on this principle. That principle is called the principle of the complementarity. Okay, so now on this basis you can find. So now how much long sequence you should have? What should be the length of the sequence? Whether it should be just a 5 nucleotide, 10 nucleotide, 20 nucleotide, 4, whatever. So for this, the principle or of designing it, all these principles depend upon the principle of probability of finding. So for that, how much length you are going to use, what the probability of that finding, what the specificity is going to be there, and when you want to, we don't want any non-specific thing. So that so the specificity, as you remember when you're talking about in case of your restriction, what we said? We said 4 raised to power 4. The nucleotide are 4 nucleotide long, then the probability of finding 256 is there. So if the 256 bases are there, then the probability of finding a 4 sequence is there. So, but here you want to be more specific. So more specific means if you increase the size of the your bases. So like you can say 4 are the bases and 5 sequence, and more probability change in 6, 7, 8, 9, 10, so on. So we want that 1 in your genome, the sequence is only once. So we design a length so that very long, that the probability of non-specific is going to be decreased. So our probe and primer, both the length of the probe and primer also determined by this formula, that the 4 raised to power n. N is the number of nucleotide long, how much long sequence you need. Okay, so So the probe is also the same, and your sequence is a complementary sequence, nucleic acid fragment of DNA, RNA of variable length. Usually, here the length is going to be 100 to 10,000 bases long. So here the term I'm using bases. I'm not using term base pair because it is not pair. It is a individual bases are there. Okay. So now this is to this you are going to call it the probe. So if you decrease the chain if nucleotide length, and we are not going to use for that way of detection, but we want to use it for a DNA amplification, or that we talk in terms of PCR, DNA replication. So then you can, if you're utilizing this sequence, this complementary sequence for your PCR or your polymerase chain reaction or your DNA replication, then we call it as a primer. Okay, so primer is going to be a short single-stranded nucleotide acid utilized by all living organisms initiation of DNA synthesis. This is the same thing that you're going to utilize. So what characteristics you're going to need here? You need here 3' OH, and length is going to be around 18 to 24. So here the length is not going to be very large as you're talking about in the case of your probes. Probes are going to be big, larger in size, because in the case of probes you want to increase the specificity. Okay, so this is the two things that we mentioned. One is the probe, and second is going to be the primer. So before moving in details, please let me know, is it clear what you mean the probe and the primer? Because if this term is not being clear to you, then moving further is not going to be much useful. Is it clear what is a probe and primer?
Yes, sir.
Okay, so this is a probe and primer. So now, as when I mentioned that the main aim of these probes and primers, both are the detection. You want to detect the presence and absence of that sequence. But as I mentioned, we are DNA. For DNA, we are blind. We can't see by our eyes. So we need a differentiating thing so you can differentiate them. So to see them, we need anything that can come up with the light. So for that, when we're talking about the electrophoresis in case of our practical lab, we mentioned that if you add a fluorophore which intercalate with the DNA, and then you intercalate with the DNA, then fluorophore is going to emit light when you are going to, it is going to absorb some radiation, it will emit some lower energy radiation. Like if you give a UV radiation in case of your ETBr, ethylene bromide, it is going to emit out orange radiation. So, so this is the one thing. So you have to link your probe. So probe is a detecting agent. So you are detecting your sequence by the use of this probe. So to this probe we can label. So to that we call the labeling of the probe. So probe labeling. So probe labeling, when we mentioned probe labeling, is you are labeling a single stranded sequence. So you are not a double strand one strand only we need to label. So here is not a type of both the strands are going to be required for the multiplication. Only one strand you need for the multiplication. Okay, so what to do? So thing is that labeling is done by mainly three different ways. One is the radioactive. So radioactive means any atom that is going to be of a radioactive nature. So a radioactive substance are those substances which spontaneously emit radiation. Non-radioactive substance, they do not emit the radiation. So if your probe that we are referring, this is your probe. If your probe is labeled with the radioactivity, so it means any of the nucleotide like TAGC is there. If you exchange this normal nucleotide with a radioactive nucleotide, so to that it means net nucleotide is going to be labeled. So simply we can say this T. If this T is going to be labeled, so T is a thymine. If this thymine is going to be labeled, so wherever this T is going to be, this become a label. So what is going to happen in this case? So now this T become labeled, and then its T is going to move here, then this T is labeled here. And if this you are moving here, nothing is going to occur, so this is even free. So if you wash it off, then this probe will, this complementary sequence, they will be washed off. But here if you are washing, they are not going to be washed off. So they will stay. So from here, if this nuclide is going to be labeled, then it is going to give you the radioactivity radiation. Means if you put a photographic film, as in the case of the, if you remember how this radioactive being discovered, this radioactive being discovered by your Becquerel or who discovered the radioactivity? Who is the father of radioactivity? Who is the father of radioactivity? Who is the father of radioactivity? Manaswini?
Sir,
Radioactivity?
sir, I'm not sure.
Hmm?
Sir, I'm not sure.
But just give a rough idea. Hmm? थोड़ा तेज बोलो.
Sir, Henry Becquerel.
Yes, Henry Becquerel is the father of radioactivity. So what when Henry Becquerel was doing, he was working with the radium. I think he put this sample over a photographic film, and when he came back, he found that this film is got destroyed. Then he wondered how this film got destroyed. So then he came to some radiation being emitted out by the crystal of this radium that he was working with. Let's lead to the discovery of your radioactivity. However, it was serendipity. Then, but further, his observation, his conclusion made him the father of the radioactivity or radiology. Okay, so this photographic film, if you put over your DNA sample, which is to which your probes are bound, then the impression of your DNA will come over the photographic film. It means if the impression is coming over the photographic film, it means probe bound to the sequence, and the sequence are present in your sample. So this is the one. And second way, radioactive, we know that it's not good to work in the radioactivity because they are the carcinogenic in nature. So there's every time there's threat of cancer if you're working with the radioactivity. So for that, what to do? We need an alternate. So the alternate is going to be fluorescence. So fluorescence is uh, your fluorescently labeled, like you talked about the ETBR that we mentioned. So in this case, ETBR, you cannot differentiate this one and this one. So, but we need a fluorophore which can differentiate. So what we are going to do, instead of this, we are labeling this with the radioactive one. At any of this end, we are adding a fluorophore. Fluorophore simply means FITC, fluoroisothiocyanate. This FITC is a fluorophore, or rhodamine, or Psi3. All and these fluorophores are there. I think all of you are aware about this fluorophore. You have studied in your chemistry classes. So if we add this fluorophore to it, then if this fluorophore is going to bind this fluorophore with your this probe with the fluorophore is going to bind here, then it will fluoresce. Whereas in this case, there is going to be no fluorescence. Okay, so this is a way how the fluorescent we are going to utilize. So this type of things we call the fluorescent labeling, or previously we called the radio labeling if you are labeling it. And another one is going to the biotrial related. It is another tag, streptavidin, streptavidin. So using a tag, if you add the streptavidin, then it's going to bind to your DNA. Then you can also detect. So, but in our syllabus, in our course, we have to talk about the radio labeling. So, radio labeling means any one of our four nucleotides going to be radio labeled. So radioactive label that you know that your phosphorus, sulfur, carbon, hydrogen, any one of these two can be radio labeled. So will this radio label I will talk a bit later. So detailing I will tell you how to radio label your probe, because when you synthesize our probe we do not they are not labeled one, because if they are labeled, radioactivity is auto decay. So each and every time they are going to be decaying themselves. You don't want that. So whenever your experiment is required, at that time we label it. Okay. So thing that we are talking about, the hybridization that I mentioned, the probe, the utilization of probe, and as I mentioned, they be based on the complementarity. So term hybridization is also there, then that is going to utilize in case of our blotting or sudden hybridization. As you talk sudden hybridization, the term hybridization. Hybrid means, as in case of your genetics or as in your normal language also, hybrid when you take mixture of two. In case of our DNA, the term hybridization we are going to utilize the complementary base pairing. Complementary base pairing of two polynucleotide chain that we call the hybridization. Or simply you can call it the formation of duplex between two complementary DNA sequence. We are going to call it as a hybridization. So hybridization can be of two types. One, you are having a double-stranded DNA. You make a double-stranded DNA single-stranded, and then allow it to form double-stranded. So that original one may become double-stranded, or a chimeric DNA strand may also form a duplex. So that is also, but when it is forming a chimeric duplex, then we call the hybridization. Okay. So for that, what we to understanding the hybridization, we also need to understand one more phenomena that is called the denaturation and the renaturation. This denaturation and renaturation we are also going to utilize for describing our PCR also, because we our syllabus also talk about the PCR. So how this PCR is going to work and how we are going to utilize our things in PCR. Okay, so what happened when we all know that our DNA is double stranded, and this double stranded is held together by the hydrogen bond. So hydrogen bond is there. So what happen if we start heating it up? So when we start heating it up, these double strand bonds, they start getting broken up. So you can see here. So in the first case, it is a double stranded DNA. So both a nicely paired one. So this is double. Two colors are there. One is blue color, second is a green color. So these blue and green are their paired one. So what happened when you denature it, when you heat it up? So these hydrogen bond got broken up, and this blue green got separated. So these blue green, they are separated one. So there are no hydrogen bond. So they are apart. So this is a single-stranded DNA, and this single-stranded formation of single-stranded DNA from double-stranded DNA, we call term denaturation. Okay, and denaturation, we know that if we heat our DNA at 94 degrees Celsius, any of the DNA, all DNA got denatured. Double from the double stranded. But this denaturation also depends upon the chain length, how much chain length you are going to have, because the chain length is going to determine how much sequence are going to be there, how much GC is there. GC is going to contribute more to this binding because it has 3 hydrogen bond, AT have 2 hydrogen bond. So GC is going to be stronger and AT is going to be a bit weaker. So they are going to contribute at what temperature they are going to. So this is the denaturation, a process of formation of single-stranded from the double-stranded. If you do a reverse thing, means you are having this single-stranded one, but the complementary sequence are present there, and you allow it to cool down, then they will renature. So denature is the reverse formation of duplex. So this process of double-stranded to single-stranded and single-stranded to double-stranded we are going to utilize in our previous thing that we are talking about, the probing or talk the hybridization. So in this process, this double-stranded, single-stranded DNA, in this you are going to mix a probe, and this is going to be a probe. So if this sequence is going to present here, then what is going to do? This probe is going to go. This probe being smaller in size, it will move and incorporate in between. Okay, so this will won't allow this duplex to complete fast, then it will move inserted in between. So if this got inserted in between, then if it is a radio labeled, it will give the radioactivity, fluorescent labeled, it will give the fluorescence activity. At night it is so biotin and avidin, separately if you move, then they're going to show a respective signal. So as I mentioned, one more thing here that it's that you called that we mentioned here, that temperature. So temperature play a very crucial role in this process, because if your temperature is very high, So very high temperature will not allow the formation of this double bond. Whereas low temperature allow renaturation, high temperature for the denaturation. So high, and here is going to be low. Okay, so this high low. So one more thing, one more concept here came. Might be we'll talk about that in when we're talking about PCR, or we are going to talk about primer. So that concept is called TM. This TM is called temperature of melting. Okay, temperature of melting is called the TM. So this TM is, as you know, the term melting. So what do you mean by melting? What is a melting? What is melting?
Sir, it's the heating of a substance so that it goes from solid to liquid form.
Yes, it is a temperature at which solid turns into liquid. Simple term. Simple our definition. If we didn't complicate it, then the conversion of solid into liquid is called melting temperature, at which temperature your solid turns to the liquid. So like for water, 0 degrees Celsius, we said it's the melting temperature of water. So at 0 degrees Celsius, ice turn to the your liquid. Similarly, different liquids have different melting temperature. Here also we utilize similarly. Here we are giving term melting to the conversion of double standard. So to double standard we can see the solid type of thing to the single standard. It's a temperature at which, or not at which, above which your DNA got denatured. That temperature is called the temperature of melting. But when we define it, for definition, we are going to define it in a different way. So temperature of melting is a temperature at which. So when you're defining, we are not defining in this the forward direction. We are defining it in the reverse direction. In this direction, so it means in renaturation form. So, but by definition or by the understanding thing, temperature of melting is the same as in case of melting. We are referring to the temperature at which your above which your double stranded DNA come up to the single stranded. So if your temperature is below 10, if it is below temperature of melting, then it is going to be more annealing. It is going to be a double stranded one. So, but when we define the temperature of melting, the temperature of melting is a temperature at which half of your DNA strand got renatured. Okay, so how we are defining it? We are defining it at a reverse way. Means it is a temperature at which half of your DNA strands they got renatured. That temperature we are calling it as a temperature of melting. Means if you say that, as we mentioned, the 94 degrees Celsius is a denaturing temperature. So at this temperature, every DNA is going to denature. But if your DNA is going to be smaller in size, so this DNA is small size, that for this you don't need to go to the 94 degrees Celsius. It may denature at in a lower temperature also. But that temperature depends upon the what GC content it is going to have. Okay, so for that there is a formula also to calculate what is going to be the temperature of melting. So this temperature formula Tm is going to be 2 into A plus T and plus 4 into G plus C. Okay, so this is a simple formula whereby you can calculate what is the temperature melting of your DNA. So in it, what you are going to do in it, like your any sequence is going to be there. Sorry, just to check. Sorry, I told you bit of this temperature melting. We'll talk detail in your PCR because there you are going to utilize it more. But you should consider one thing. Uh, at the temperature melting, your DNA is going to be a single stranded more than the double stranded one. Above the temperature melting, your DNA is going to remain single stranded. It's not going to be a double stranded. Okay, so thing is that what we are saying, Temperature determines the pairing. If you are having a sequence at a higher temperature, then any DNA sequence whose temperature of melting is going to be lower, they won't pair. Means you can simply also can say that like this is a long like this is sequence that you are having, and this is a probe that we are having. So now thing is that over this long DNA strand, there is only one spot where this sequence is present. But as we mentioned that all are ATGC, so somewhere you found that like at this end 80 is there. But this end may pair where anywhere like this way. At this end 80 is there. Like if some end here also some sequence is there, it may pair like this also. So where the 100% complementarity is there, pairing is going to be like this, and where less complementarity, the pairing may be like this also. So what we want to do here? So we want to avoid this. Sorry, we don't want in our experiment. Sorry, just a moment. In our experiment, we don't want this type of pairing or this type of pairing, or you may say that this type of pairing. Only what we need this one. So how to avoid this? This we can avoid by temperature. This is small region, very less amount of hydrogen bond is there, very less amount of hydrogen bond will be there, very less, but a stronger one. So more hydrogen bonds are there. So if you heat it up, so you keep a temperature higher. So at higher temperature, non-specific hydrogen bond formation won't occur. This won't occur, won't occur. Only such type of thing is going to occur. So this temperature is going to help us in this way. There is renaturation that we are talking about, because this renaturation is going to help us in in hybridization. Because when something is going to renature, then only it form duplex. And the what type of duplex we need? We need a chimeric duplex. Okay. So these are two things that these are phenomena on which your hybridization or the bloating or the southern hybridization or southern bloating that we are mentioning, they depend. Okay, so now one more thing is there that we talk when we are talking about uh, when we are working, when we perform such type of the experiment, this hybridization experiment. So in this hybridization experiment, we need to perform over the DNA. So, but we all know that DNA is present within the cell, and it is very difficult to work within the cell, and so that what we need, we are extracting our DNA, and DNA when we run the gel, then it is going to be within the gel. Then inside the gel you have to when you add your probe to bind it, then the gel won't allow to enter your DNA to it so that it can bind it. So we need a system where a DNA is going to be present on the surface, because any substance, anything is going to present on the surface is exposed one. So like simply in case of COVID also, when we say that stay inside the home, because inside home the chances of virus movement is going to be very little. If we are going out, then you are going to be more exposed to the virus. So similarly here also, if you want your sample to get exposed with a probe, then they should be present on the surface. But in case of when we are analyzing a DNA sample, how we are analyzing? We are analyzing on the gel. Whether you are when you are cutting with the restriction endonuclease is also a restriction endonuclease to produce fragments. So large number of fragments are going to be there. So all those fragments will also be analyzed on the gel. So your DNA, then you perform our analysis is generally within the gel, because in the gel you can recognize where this segment is present, of what size this segment is going to be there, and it is easy to handle also. So to work over it, so we need a solid sport. So what type of the solid sport is going to be this? This solid sport is going to be there to provide a support so that your DNA or RNA is going to be immobilized. No migration is going to be there. So in case of solution, the diffusion is definitely going to be, the particles are going to move here and there. But if it is going to be on a solid spot, it will stick there, it won't move here and there. So no intermixing is going to be there. So in case of your gel, if you remove the electric field, then the normal diffusion, also your DNA start migrating three-dimensionally. To avoid that, we want to immobilize our DNA RNA over a solid spot. The property of the solid spot should be, it must be inert. Inert means, when we are saying inert doesn't mean it does not have any property. Inert means it must be chemically inert to our DNA. But as I mentioned, interaction is going to be there. It must interact. Otherwise, how it is going to bound? Okay. So second thing is when it is bound to a surface, these bound sequence, they are available for hybridization when added the nucleic acid probe. So when we add a nucleic acid probe, these DNA sequences are available. So that's why we need a solid spot, or transferring your DNA or RNA over a solid spot is called the transfer or blotting. So term DNA blotting, then what we're talking about, like southern blotting, northern blotting, these all things depends on where we are transferring. Okay, so two types of the solid spots are there when you are transferring. One type of solid spot is your filter hybridization, or your paper hybridization, means your filter paper we are going to refer. If you transfer your DNA, RNA or protein over a filter paper, then we use term filter hybridization. Depending upon the name, DNA we use the southern blot, RNA we call the northern blot, protein we call the western blot. Okay, and the second type of solid spot is in silica. Silica is, that your glass slides. So glass slides are made up of the silica or borosil, that is borosilane. So the silica is there in it, your glass. So if you are transferring, you are immobilizing your DNA or a silica glass slides, then we use term in silica hybridization. This in silica hybridization is of different type. The microarray technology that you might know, that's also in silica. Over a slide, you have transferred a large number of design large number of wells or large number of fragment you have transferred, and you know that each and every location what sequence is present. So this microarray is also a type of hybridization, and also called the fish fluorescence in situ hybridization. When you are working with the fluorescence within your cell, then you call the in situ hybridization. If you are working within a tissue or within a cell. And the fish is also a type of in situ hybridization. Fish means after stand for the fluorescence in situ hybridization. So this is some basics of things. So now thing is that how you have to blot, how to transfer, what type of the setup you need, what are the different things that you need. So first thing, when you start with the blotting, so this blotting is going to be, as the name is there, by the Kemper reaction or the southern blotting, which is described by the Southern in 1975. So on that basis we name it, and how it is being transferred, what principle is going to be transferred. We are going to utilize electrophoresis for this transfer, or we are going to utilize a capillary reaction. That's different techniques they utilize differently. So if you want to see the how the southern blotting is we need to do, we have a southern blotting instrument at our laboratory also. It is if anyone of you visited Dr. Sunny lab, you might have seen a large chamber is present in his lab. So this is a hybridization chamber, and a rotatory instrument is also there. That is also that's also that we call the that is for the baking. Different type of things are there that will in detail we'll take in our coming class. So what type of things, what type of setup is there? You can see the one negative and positive electrodes is there. If you want to utilize electricity, then in between you produce a gel holder, a sponge pad is there, filter paper is there, then transfer paper is there, then gel is there, then again filter paper, then sponges, pads, these things. So you can see that one surface of your gel is exposed to the your transfer paper. So this transfer paper is that solid spot that we are referring. And this transfer paper is based on a filter paper based, or these filter papers are not just these transfer paper and the filter paper that is written here. They differ in what type of the paper that you are going to use. Okay, so all these in detail we'll talk in our next class how these things are going to perform and what different types of the membrane are going to use. So mainly you are going to utilize 3-4 type of membrane. One is a nitrocellulose membrane, a nylon membrane, DEAE membrane, CM paper, also called the PVDF membrane. So these all we utilize and all have the merits and demerits. In detail we'll talk about in our next class. So now we are at the end of the session, and so please turn on your camera. Before that attendance, if you have any query what we have talked till now, please let me know. Any query you have?
No, sir.
Okay, so if no query, so please turn on your camera when I call your names. Just a moment, I am coming on your attendance sheet. Okay, so who will tell me who came late today? Who came late today? Kshitij? Kshitij?
Yes, sir.
At what time you joined?
Sir, I think सर मैंने 4:25 के करीब ज्वाइन कर लिया.
Join at 4:25.
सर वो कुछ काम आ गए थे इसलिए.
तुम सोचते हो
No,
मुझे
but
पता है
सर वैसे मैं सर ऑन टाइम ही ज्वाइन कर लिया.
फिर मैंने पूछा जब क्यों नहीं बताया तुमने? When I was asking who joined late, why didn't answer that?
Sorry, sir.
And what happened in your last class? What you did in the last class? Kshitij? What you did in the last class? Kshitij, I'm asking something to you.
सर तो याद नहीं आ रहा.
You left the class as I done the attendance in the start. हो गई चलो.
सर वो
हाँ सर क्या हुआ? बताओ.
सर नेटवर्क इशू आ गया होगा सर क्योंकि मेरे
आ गया होगा
क्लास
कि क्लास बंद करके सो गए थे. नेटवर्क इशू आया नहीं आया क्या रहा क्या नहीं रहा. आ गया होगा. मैं शुरू से लेके एंड तक नेटवर्क इशू ही रहा और तुमने देखा ही नहीं कि नेटवर्क इशू है.
नहीं सर ऐसा नहीं है वो सर वो बीच बीच में कभी वो कट जाता है वो ऐसे सर मैं
MS टीम सब बताती है कब कब बढ़ता है कब कब जुड़ता है. समझे? ये इलेक्ट्रॉनिक सिस्टम है यहाँ.
ध्यान रखूंगा.
ये मतलब कैमरा ऑफ सब यहाँ दिखता नहीं है तो भी दिखता है. सुधर जाओ. ठीक है. ओके
सर अटेंटिव रहता हूँ.
अटेंडिव तो मुझे पता है कितना रहता है. आदेश आदेश
प्रेजेंट सर.
अतोयबा
Present, sir.
आयुषी
Present, sir.
Demetrius
Present.
Devanshi? Devanshi? Ishita?
Present, sir.
Janvi?
Present, sir.
Kshitij? Manaswini?
Present, sir.
Present, sir.
Kshitij, turn on your camera. Namrata?
Present, sir.
Nehal?
Present, sir.
Upal?
Present, sir.
Saiba?
Present, sir.
Sanjana?
Present, sir.
Shakti?
Present, sir.
Tanvi?
Present, sir.
Tridev?
Present, sir.
Vivek? Vivek?
Present, sir.
Yukta?
Present, sir.
Yuvraj?
Present, sir.
So thank you all. Stay safe, stay happy. Good day. See you again tomorrow for your laboratory session.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.